Hello, my name is Mark Temple, and this is a mini lecture on cancer genetics. And in this particular lecture, we're looking at the multi-step tumor formation. So what we're discussing in this lecture is a series of events that occur within a cell that give rise to a tumor cell. So these genetic changes that occur in the cell proceed over many years, often decades, and they give the cell a different phenotype, a different growth characteristic over the normal cell, which tend to give it the ability to grow more and to cause problems in the body. So cancer normally originates in proliferating cells. So it's those cells in the body that are dividing rapidly. And when a mutation occurs in these cells, the daughter cells contain that mutation, so that you get a series of a generation of cells that contain that mutation. And then when those cells pick up a second mutation, they pass it on to their daughters, so that the daughters have now got two mutations. And then when a mutation occurs in one of these cells, you get more mutations, and it's a flow-on effect. This doesn't occur so much in cells that aren't dividing rapidly, such as neurons and muscle cells, and therefore you don't tend to get tumors in these tissues as frequently as you do in the rapidly dividing tissues. So what is it that drives tumorigenesis? Effectively, it's a series of genetic mutations or epigenetic alterations. So these are mutations in the DNA that affect coding sequences or promoter regions that affect gene expression levels. So this gives rise to different amounts of proteins in the cell, and it gives rise to proteins with different sequences, and therefore these proteins are not able to function or function hyperactively. Now, these mutations are occurring throughout the genome, and often they don't cause problems. But there are particular genes in the genome that are involved in controlling cell proliferation, cell survival, and other really important phenotypes. And when you get mutations in these cell types, they're the ones that are going to drive tumorigenesis. So people who study cancer have been looking at the epidemiology of cancer. In other words, looking at large populations and the statistics of these populations, and looking at the behavior of cancers within these populations. So one of the things that's pretty obvious to us all is that cancers are observed more frequently in older people than younger people. So, studies have shown that a 70-year-old is 1,000 times more likely to have cancer than a 10-year-old. Okay. Now, this is because it takes time for these mutations to pick up and be passed on to daughter cells, and then to accumulate in this series of mutations. Now, another important insight into the occurrence of tumors is that it's not simply related to age. So if you're exposed to a chemical that modifies your DNA, such as smoking causes DNA damage, or sunlight causes DNA damage, then there's a period of time from this exposure and repeated exposures that leads to the onset of the disease. So there's a graph here that shows for different age group. So if you're 15 to 24, or if you're 35 plus, depending on your age, if you're exposed to a carcinogen, whether you're 15 or whether you're 35, then you have to wait a period of time before the accumulation of mutations through these series of exposures leads to a cancer. So yes, cancer is more prevalent in older people because they've been exposed to more genetic mutations over their life. But if you're exposed to a particularly nasty carcinogen, then there'll be, effectively, it doesn't matter whether you're a 35-year-old or a 15-year-old, there's going to be an incubation time before that cancer occurs. So this repeated exposure causing repeated damage to these cells, accumulating damage in cells, giving rise to the tumor. So different genes are involved in different cancer types, okay? And different tissue have different susceptibility to cancer types. But we're going to be looking at some of the lessons learned from colon cancer, and some of the reasons why colon cancer is well studied and fairly easy to study is that you can undergo a colonoscopy and you can look at the cell lining of the colon in the same people during their lifespan, and you can look at the progression and changes that occur within the lining of the colon, and you can take samples of these different cancer or early cancer or late cancer cells, and you can look at genetic markers and look at what's happening in these cell types in the patient. People look at the histology of these cells under the microscope. They look at the rate of division of these things, and you can see that you see a thickening of the lining of these of the colon. And some in later stage cancers, you can see that the cells under the microscope are no longer well organized. They're sort of looking that they've lost their characteristics of normal cells, and as the progression of the disease occurs, then these are referred to as polyps or as adenoma. These are fairly benign unless they're able to break through the basal membrane and then invade the bloodstream or the vascular system, and then be transported around the body, where they may invade other tissues and become malignant. So when we're looking at colon cancer, it became apparent that the tumor development is a series of phenotypes occurring in the same tissue. Okay, so there's two schools of thought at the time. One is that a normal cell can undergo a mutation, giving rise to a highly malignant cell. The other theory was that a normal cell undergoes a small change, giving rise to a cell with a slightly different phenotype. Within those cells, a second mutation occurs, giving rise to faster growth phenotypes, and a succession of mutations occur in this dividing cell, or in these dividing cells, giving rise to the cancer phenotype, rather than a normal cell becoming cancerous almost instantly through a mutation. It's a series of mutations in these dividing cells. And there's 3 bits of evidence that indicate that this succession of tissue phenotypes is actually what happens. So one of these pieces of information shown here is that within these cells here that have picked up some mutations. So these are the normal cells. These cells have picked up mutations to become an adenoma, and then from these adenoma cells, the malignant cells are growing. So these are your normal cells here, showing the normal sort of phenotype here. Here's the adenoma, with showing a different phenotype here, and then from these cells is given rise to the malignant cells here, which have the ability then to grow even faster and start to metastasize. You can see how there's a succession of phenotypes occurring in these cells. So another piece of evidence that suggests it's a succession of phenotypes is that if you actually remove the polyps from the patient, then you reduce the risk of cancer by about 80%. So as shown here, in patients who have undergone a polypectomy, as opposed to those who haven't, these polyps, these patients can have the polyps in their colon, and they have a higher rate of cancer, indicating that the polyps are migrating to cancer. And if you remove the polyps, then you remove that migration to cancer. Okay, so if it were normal cells not going through the polyp stage but going to a malignant stage, then removing the polyps wouldn't have any effect. But this shows that when you remove the polyps, you reduce the incidence of cancer, indicating that normal cells go to the polyp stage, which then go to the cancer stage. So that again is strong evidence that it's a succession of phenotypes. And the third piece of evidence that indicates that it's a succession of phenotypes it comes from familiar studies. So if you've got a family history of bowel cancer if the genetics of your family history indicate you're going to have a higher rate of colon cancer, then people have done studies on these patients and identified a mutation in the APC tumor suppressor gene. So there's a particular mutation that is inherited in these families. Now, this mutation gives rise to the formation of lots of polyps in the colon. So hence the name familiar adenomatous polyposis, FAP. Doesn't matter what it's called. But these patients have a mutation in the APC tumor suppressor gene, and that gives rise to the polyps. Lots of polyps, you can observe them, and in these patients they have a higher rate of colon cancer arising from the polyps. So this is showing that again, there's a succession of changes. The first change apparently is a change in the APC gene, giving rise to the polyps, and then within these polyps, within these cells that have picked up that have this mutation, they pick up additional mutations that become cancerous. So if this is true for colon cancer, then it's probably going to be true for other cancers as well, where for other cancer types in the body, there's a succession of genetic mutations that occur in fast dividing cells, and the buildup of these mutations gives rise to cells with uncontrolled proliferation. So I've got a nice series of pictures here that show this idea of a succession of tissue phenotypes. So here we have a normal cell, and here we have a cancer cell, and it's not as if a mutation occurs in one of these cells to give rise to a highly malignant cell type which then grows. It's more a case of, in a normal cell type here, you can see these cells are well organized, they're in close proximity to each other, and but at some point in these normal somatic cells you can pick up a mutation in one of these cells, and typically this is at a tumor suppressor such as the APC gene, and this gives this particular cell a higher rate of growth than the surrounding cells. So this cell will outgrow these cells. So after a period of time, maybe a few years, then that cell has divided again and again and again to give an area of that tissue that has many thousands of copies of these cells, all with that same mutation, because they're derived from each other. And then one of these cells at some point in time picks up another genetic mutation that gives it a faster growth characteristic. So then it outcompetes the cells around it to give a succession of phenotypes. This is another phenotype that's now occurring. Something you can see down the microscope. The cells are starting to look different shape, they're starting to be less well organized, and one of these cells now picks up a mutation, and you're getting these succession of phenotypes that it can be observed during the progression of the disease. So at some point these cells are growing rapidly and are able to metastasize and leave this particular area of the body into the vascular system, and then there's a chance that another tissue in the body that's receptive to these cells can absorb, not absorb, they can pass through the vascular system, and then they can start to grow in maybe the liver or another tissue. So this has been observed for colon cancer, and over say a 30-year period, something like that, you get the normal phenotype progressing through a series of mutations in these same cells to give a cancerous phenotype. And this is also thought to be true for other cancers that they've looked at, such as head and neck cancer, cervical, lung, breast, and prostate cancer. So again, there's that they've identified some characteristics of these that follow this idea of a succession of phenotypes giving rise to a cancer cell. So effectively, there's a series of progressions going from a normal epithelial cell through to these more hyperplastic epithelial cells, and then you get these adenomas forming, and they can be observed as, classified as early, intermediate, or late, leading to carcinomas, and effectively then you're getting these malignant tissues that can leave the colon, the lining of the colon, into the vascular system, and then can be absorbed by other tissues. So we're saying that it's a multi-hit hypothesis. We're saying there's a multi-step theory of tumor formation. So what are these multiple steps that are occurring? So some of them, one of the first genetic events that we know about from familiar studies is the loss of the APC gene. So this is a tumor suppressor that can be lost. And this is one of the first events that often occur in the progression of a normal cell into a cell with starting to move along that tumor progression. And then we also talked about, genetic mutations and epigenetic mutations. Now, some of the other things that have been observed in tissue samples from these patients is DNA hypomethylation. So you get methyl groups added to the cytosine residues in the promoter, changing the expression of that gene. Again, as you progress through the tumor formation, you see activation of oncogenes, such as a mutation in the RAS oncogene, and also there are other tumor suppressor genes that get mutated as the cancer progresses. So when you look at these tissues and you do genetic analysis of these tissues, you can identify particular mutations that occur. Some that occur in early stage tumor formation, some that are more common in late stage tumor formation. And again, there's a nice diagram here that shows this idea of a succession of phenotypes, but it's not always the same succession of phenotypes. It can be different phenotypes, different genes, different orders of mutations, but it's a series of mutations that give you that tumor phenotype. So a typical example might be a loss of the APC tumor suppressor, and then an activation of the RAS gene, which is an oncogene. We'll look at RAS in a later lecture. It might be some other gene that hasn't been characterized that's involved, and then it might be a mutation in p53. So as you go from a normal cell through to a malignant cell, this might be some of the key players involved. Likewise, it might be a mutation in the RAF protein rather than the RAS protein that occurs in some patients or in some cell types. And then following on from that, it might be this other gene referred to here as DPC4, which is, I think, one of the SMAD proteins. And then this might be followed by some other uncharacterized genetic mutations giving rise to a cancer. It might be that there's a mutation following from APC in the PI3 kinase, the phosphoinositol 3 kinase protein, which phosphorylates inositol on the cell membrane, leading to the activation of a whole series of kinases. It might be mutations in that gene that are important, and then that might be followed up by another mutation that's not characterized, and then that might be followed up by a mutation in p53. So that might be the genetic pathway. It might be a series of uncharacterized pathways in some patients, or it might be TK mutation. TK is a tyrosine kinase receptor on the membrane of the cell. So if you have a mutation in one of these tyrosine kinase receptors, this might be an early stage mutation following from the APC mutation, and then that might be followed by one of these SMAD or DPC4 mutations. So it might be that in different patients there's a different series of genetic events that give rise to a more malignant phenotype. And this is just showing some of the key players and some of their involvement. Okay, thank you for listening. I hope this has been of some use to you, and